Hello everyone. Um, Today I'm going to talk about a growing field, which is cave microbiology. And here we're studying Bacillus genus isolated from different caves in Algeria. This is the presentation plan. So first of all, why Bacillus? So Bacillus is one of the most diverse and commercially useful groups of microorganisms. It is widely distributed in soil, air, water, and caves due to their metabolic diversity and their low reported incidence of pathogenicity. Bacillus has been used in a wide range of industrial processes. On the other hand, Bacillus species had been used as probiotics for at least 50 years within the Italian product Enterogermina, which is known as an OTC medicinal supplement. So those are some of Bacillus used as probiotics, like Bacillus subtilis, Lichenoformis, Cereus, Pimelis, closey, and coagulants also. So this current work, it seeks to contribute in finding some biological solution for digestive metabolic problems and the viralization of cave bacillus. But why caves? So cave microbiology is a growing interdisciplinary field which involves microbiologists, geologists, and chemists. Caves are characterized by extremes of near starvation or oligotrophy, which promoted a big potential importance. They have less than 2 milligrams of TOC per liter, and only one study had been done in Algeria about cave microbiology. So in Algeria, there are different cave types according to the geology of the rock and also to the geometry of the cave. So we can find sandstone, carbonates, lava tube, gypsum, dolomite, and according to the geometry, horizontal, vertical, and mines. So now I'm going to present some of the sampled caves in our study. So the first one is in the Algerian desert, and it's a sandstone cave in a city called Mshunish, Biskra department. So for each cave, we studied different parameters, physicochemical analysis temperature, humidity, and pH. And samples were cave sediments, or if we can say a cave soil. We noted also the presence or the absence of bats and other animals, considering it as a source of organic matter. Um, so the second cave was Ghardba in Constantine department, which is a karstic cave and which also contains the biggest number of bats among the other sampled caves in our study. So also a big quantity of excrement, bat excrement. So in this cave also we studied excrement. The third one is a mine, Ain Smara, used before for extraction of onyx, and it's also in the Constantine department. Then we have Anubuswil. It's in the Georgia mountain in Buira department, and it's the second deepest cave in Africa, and it's one of the most studied geologically among Algerian caves. So in this cave, we had 4 different samples. One is in minus 50, minus 100, minus 200, and the deepest one is minus 450 in the fossil network. Then we have Pirate and La Blanche in Bijaya department. La Blanche means the white, and because it's full of crystals and also helictite. So the other one is Rajma in Gelma department, and it's a clay cave. And finally, Yucus in the Tibissa department, and it is known by its folding water. So as you can see, we try to sample different different cave types in Algeria. So all the samples were taken from the dark zone, and this is a picture explaining the three different cave zones: the entrance, twilight, and then the dark zone. So those are some of cave speleothems that or like formations that we found during samplings, like biovarmiculation, oxidations, and etc. And those are some of the animals and insects found there, like bats, spiders, myriapods, and so many others. And now with the lab part, or the lab analysis. So first of all, we did pedological studies, which is organic matter, conductivity, lime requirement, etc. Then isolation and purification of these isolates or strains, and then the enzymatic study, safety assessment, and probiotic potential. So first of all, we proceeded the heating treatment to eliminate all vegetative cells and select only spores. And to avoid Clostridium spores, we prepared aerobic cultures. And it is really important to note that the heating treatment do not induce sporulation. It will only select spores that already exist in the soil or in the environment. So those are normal pictures about isolation. And then we did the microscopic observation to check the cell structure, which is obviously, as you see, bacillus. So after that, we studied the enzymatic potential, which is digestion of gliadin. So for digestive enzymes, there is like different type, and the one that we're studying in this case, it's gliadinase. So before we talk about the enzyme, let's first explain the syndrome, which is gluten intolerance, and which is a digestive metabolic disorder. It is an allergy to the degradation product of gluten, which is gliadin, and which induces an immunity reaction. So first step was the gliadin purification by the use of 16% ethanol, incubation, centrifugation, and then lyophilization. And then to confirm the product, we processed an SDS page electrophoresis. Then we tested the enzymatic ability for our 250 isolates in solid medium and then in a liquid medium. And as you see, there is like a hydrolysis in the solid medium. So, and then we studied the safety of these isolated strains. So we did first a lecithinase test, which is with using a yolk agar, and then hemolysis activity using Luria brethani blood agar, and finally MYP agar or Bacillus cereus agar. And those are some of the results. And finally, an antibiogram. Then the antibiotics was like selected according to inhibition of protein synthesis like amikacin, erythromycin, inhibition of cell wall synthesis like oxacillin, cefazolin, and finally inhibition of RNA polymerase action like rifampicin. So, among the 250 isolates, only 16 had been chosen for their safety aspect and their enzymatic potential. After that, some of the probiotic aspects have been studied with these 16 uh, strains. For example, growth at 37 degrees Celsius and the resistance to acidic pH. And finally, sporulation test had been performed using TSA medium, supplemented with 5% manganese sulfate. And finally, further test is considering the probiotic potentiality and the safety of selected bacillus are required, and of course, molecular identification of selected microorganisms is needed. Thank you.